Bapak-bapak dan ibu-ibu yang kami hormati, ladies and gentlemen, distinguished participants, thank you very much for coming to our event. This is our second international seminar on One Health, New Roads Towards One Health in Indonesia and the region through the No Self mechanism. I guess we are all ready to start. So may I welcome Professor Dr. Bungaran Saragih of Ecotalis, also our former Minister of Agriculture, to give us his welcome remarks. Professor Bungaran, may I have your time, please?
Selamat pagi. His Excellency, Minister of Health, or the one who represents him. He is supposed to be here, but he is now in the United States. The Honorable Chairman of National Research and Innovation Agency, Badan Riset and Inovasi Nasional, BRIN, or the one who represents him, but maybe soon he will join us. Distinguished speakers, esteemed participants, dear colleagues. Horas. I bid you all a warm welcome to Bogor and the EPB Convention Center and our One Health seminar. I appreciate your presence all the more because of the current risk that social mobility gives to exposure to the pandemic. Without doubt, today is a special occasion because I recall we had the first seminar on 28th of October 2019. This was just a few months before the outbreak of the current global pandemic. Our second seminar today takes place about 3 years into the pandemic. The pandemic challenged everyone in the world, both citizen as well as government, in their respective roles, choices, and option of response. The heart of the matter is obviously that with increased mobility, the risk of disease jumping from animal or plant to human is increasing. All have to learn how to live with the current situation with regard to health. Since we spoke about the benefit of the no-self mechanism back into 2019, the no-self mechanism has therefore assumed bigger proportion. The needs of country including Indonesia and other countries in the region, have grown. And now are many. One very clear need concern a way forward toward health for human, for animal, plant, fish, water, and the wider environment. One health is essential, and pressure especially from urban areas, and also tightening international and national supply chains and related industry will drive development. Even though globally huge public money were spent on vaccines development, globally there is also a shortage of resources devoted to sustainable and environmentally acceptable step forward. In most countries of the world, the response of the government have included the centralization of its allocation capacity to government-funded and public research. Indonesia is no exception, as the President Jokowi decree in 2019. National Research and Innovation Agency, or in Indonesia we call it Badan Riset Inovasi Nasional, BRIN, is to take the responsibility of allocation and use of government fund for research and innovation. I think that we can say that BRIN is here at the right time. Furthermore, we also notice how universities, especially in Indonesia and elsewhere in the region, are also concerned with the One Health concept. Some universities established a One Health collaboration center to involve in the study of this new paradigm. Let us be mindful that the collaboration among government institutions, research institutes, and universities will enable us to understand the complexity of the global health problem and effectively mitigate the possible causes. I personally understand that this forum is a blend of science, policy making, and business community. It should therefore be able to make use of our insight for the sake of a healthy society. Many roads will then involve the use of current and advanced methods such as molecular biology and bioecology in both agriculture, maritime, and health sectors. As a new developing approach, I understand there will be some dispute regarding potential use of it, particularly the so-called self-DNA. This forum will then could be the platform for all of us here to reach the same angle so that we could realize benefit of this new paradigm maximally. Esteemed participant and dear colleague, today we are honored by the presence of distinguished researcher from University Federico Naples, Italy, Professor Mozzolini, will share general insight in the no-self mechanism. And we will also learn from Dr. Marcelo Diano about algae and its unique role in devising application. Dr. Rija Putranto will discuss application in the oil palm industry, the biggest industry in this country. And Dr. Romy Nofrianto will present possible applications in the marine and fishery sector. I'm also grateful to Dr. Joko Pamungkas for his willingness to speak about One Health in Indonesia as a whole. I would like to personally thank Mr. Peter James, CEO of A&D Biopharma, for his continued commitment to the developing world in his endeavor to share the benefit of the no-self mechanism in a wider context. I would like to express my personal and deep appreciation to him. I hope that soon Mr. Jens will be able to travel again. He is not here. So we can welcome him here in Indonesia and the region. In Asia in general, and definitely also Indonesia, there is not a great deal of investment in research and development in agribusiness and also in pharma, I believe. This paucity can be explained historically and also by the generally very high competitiveness of Asian factor markets. It is all very understandable. There are a great many of generic products, all more or less functional for the purpose they were designed for. So the status quo and business as usual continue. But there are many signs that the efficacy of previously efficient weed deciders, such as Roundup, the famous Roundup, reduced, while disease has sprung up for which no effective remedy has been found yet. Historically, it may be so that the period of easy costing is coming to an end, because there is now a growing need for investment by agribusiness and pharma alike. Investment is part of responsible business and also very much part of good and sustainable business. I wish all of you success. I understand that the seminar has been organized to maximize interaction so as to involve you all. And I would like to invite you all to use the opportunity for sharing information. We highly appreciate your presence, and I personally would like to extend my thanks to PT Ecotalis Technology Indonesia, who organized this event. I wish you all fruitful deliberations. Thank you.
Thank you very much, Professor Dr. Bungaran Saragi, for your very warm welcome address. May I now call upon Professor Dr. Didi Hajar Gunadi, who will share with us the brief annotation on the flow of this seminar. Thank you, Pak Didi.
Bismillahirrahmanirrahim. Good morning, everyone. First of all, thank you for your coming to the second international seminar on One Health, organized by PT Ecotalis Technology Indonesia. And we, or some of us, maybe already here twice since 2019. Before COVID, we conducted the first seminar on One Health. Thank you for your coming, and we will have a program for today, starting from this morning until this afternoon. And the organizer would feel sorry about this delay. We about 30 minutes late. This is due to we arranged our VIP guests, and fortunately they both will come and join with us here. So we will have 3 presentations this morning before refreshment, I guess. And starting from the initial, what you call it? In Indonesia used to be called as a provocator. This Peter Jen. He's from A&D Biopharm, but unfortunately he could not join us here, but he will present his speak with some video. Yeah. And then we'll continue with the presentation or speech from the representative of Minister of Health, and very specifically, he said he apologized because he couldn't join this seminar because he's still in the US. And last night he told me he will send his staff here, and he will be represented by the acting head of the Agency for Health Development Policy. I don't know whether it's already here or still on the way. And it is actually she. Yeah. She will be here in a minute. And this is the new organization that used to be Agency for Research and Development for Health. Now, because they are joined with the BRIN, Agency for Research and Innovation, National Research and Innovation, then they what you call it, convert to this new agency in the Ministry of Health. And then the second speech will be the representative of the head of National Research and Innovation Agency. Dr. Handoko, also apologized for not able to join with us here because they have a launching program. They call it INARI. INARI as Innovation of Research and Innovation this morning in Jakarta. He is going to be represented by the head of Research Organization for Health. And this is also she, right? Yeah. And then after that, we will have a short question and answer, and before morning refreshment break, if time permitted. If not, the refreshment will be served on the table, I guess. Yeah. Okay, and then in the next session, we will have our outstanding guest here from University of Naples, Professor Stefano Mussolini. And we also welcome him because it is the second time he came to this room, I guess. Yeah. He's going to present the update of the One Health, or one aspect of One Health, about the evidence of the prospect of no-self technology for combating pests and disease. And then next to him will be his colleagues, Marcelo Diano. He's also coming from Italy. He's representing the M2M company. And he's going to share with us about the developing a no-self technology product using microalgae as production and carrier platform. And then short question and answer, and we'll continue with lunch break at about noon for 1 hour. The afternoon session will be presented by Dr. Risa Ariputranto, DEA. Is from the Indonesian Oil Palm Research Institute, Bogor unit. He will share with us about the potential application of no-self DNA for agriculture in Indonesia. And then the next speaker for this afternoon is Dr. Romi Novriadi. He's from Polytechnical University of Fisheries, Ministry of Maritime and Fishery in Indonesia. He will share with us about the current finding on self-DNA for fishery in Indonesia. And the last speaker for this afternoon is Dr. Joko Pamungkas. He's from Indonesia, Bogor Agricultural University, and he is responsible for Indonesia One Health University Network, or Indohan, in this university. He will share with us about the reflection on the One Health concept for animal and human diseases, or zoonosis. And then after this presentation will be some general discussion, and networking will be led by our single author or single actors, Dr. Taku William Botema. And after that, the closing will be about 3:30 p.m. and I will be adjourned this afternoon for this seminar. I think that's the agenda for us today, and I have also a function as moderator here, so I will continue my function here, sitting in the front. If the representative from the Ministry of Health already here, please come up with us here, and also from BRIN, Ibu, silakan Bu di depan, and we'll start with the video of Peter Jan first. But please come to join with me here. Okay. Could we start the video from Peter Jan? Peter Jan is the CEO of E&D Bio-Pharma.
Dear Excellencies, dear Chairman of the National Research Innovation Agency, dear Chairman, distinguished speakers, participants and colleagues, and of course also hi to Nick, my son, who represents the next generation of crazies. As Professor Bungaran said, I caught recently COVID when traveling through India, and I'm very sorry I can't attend and meet the many friends I discovered in Indonesia, as well as the new friends from Malaysia, also here in the audience. 5 years ago, I fell very ill with a persistent bacterial infection, and from then on, I really wanted to find alternatives to the antibiotics that did not seem to work anymore. And I now know that Professor Mazzoleni's DNA self-inhibition principle is just what the world is looking for when fighting AMR, or antimicrobial resistance. Also, the first meeting I had after my recovery, the first car drive I could undertake was to meet Pak Takel at a nice castle in the south of Holland. And from then on, I got totally addicted to Indonesia. Its clever people, the warm welcomes, the impressive nature, the industrious cities and the enormous potential it has, and on its way to becoming the fifth economy in the world. But it was also your motto, unity in diversity, that resonates with Professor Mazzoleni's invention, an oscillation between self and non-self, between the individual wants and the societal needs, that made me really want to commit to Indonesia and set up with Pak Tako and his impressive team the first One Health seminar. And let's face it, the Republic of Indonesia, with its neighbors united in ASEAN, is uniquely placed to become a world leader with the One Health approach, and that is what attracts me to your great countries. A young, bright research population, outward-looking, quick to unlearn the existing paradigms, reappreciating agricultural and health traditions, embracing this new and elegant finding that while for an individual organism, DNA is important within the cells, but one outside a cell, DNA plays a role in population dynamics hitherto unknown. Simply said, when an organism, any living organism, ingests or otherwise gets in touch at an early growth phase with its own extracellular DNA, it will stop growing and eventually it will wither and die. Professor Mazzoleni will explain more in detail, and Dr. Diano will let you know the ins and outs of the applications later today. And also Dr. Roman Novriadi will present a series of important in vivo trials here done in Indonesia. This new technology will offer shrimps and other patients an alternative to antibiotics and may one day safeguard society in case of a bacterial pandemic. And not in the least, of course, the technology also offers farmers a fair chance of reducing pests and plagues in their crops, affordable and accessible. So after a rocky and winding road and one viral pandemic later, here we are again. We mourn the many that had to leave us, but we can deliver the living a fully de-risked project, sufficient proof of concepts in vitro and in vivo in both health and agriculture, and this second One Health seminar. With pleasure, I have noticed that, especially in the ASEAN countries, researchers are very interested to learn and to discard old paradigms when new evidence points at new directions. And that's also very true in Indonesia. People unlearn rapidly, while in the West, big names, entire faculties and established magazines refuse to unlearn and steadfastly defend old views, sometimes to an absurd level of stupidity and to the detriment of farmers and patients. There are two paradigms I like to stress here, two new ways of looking at things. The two are different but connected, and together they will create a perfect storm, resulting in hundreds of new products and applications. Products that can be produced here by local companies for local problems and within ASEAN countries offering new export opportunities. The first point of view is that one can only work successfully in health, food and farming if all research and regulatory silos are much better connected. Research disciplines that can be connected must be connected. One Health is the new paradigm through which policies will be shaped, budgets will be allocated, and, who knows, people will vote. It is great that the Indonesian government is breaking down already the barriers and helps very different disciplines to connect and interact. The other new paradigm is about genetics. We all have been trained in the modern synthesis, basically stating that DNA is the blueprint of life. Now, another way of thinking is emerging, called biological relativity by Dr. Dennis Noble, where DNA is seen only as an organ of the cell, in a way recording and adapting to external influences. So context and environments are becoming perhaps more important than just genes and sequences. This biological relativity sounds heretic, and some in the audience will cringe. Rightfully so, as this paradigm shift will, in time, influence research budgets, reprioritize applied science efforts, and surely also increase the interest in biophysics, a field that brings novel technologies as well. So beware, a change is about to come. Indonesia, with its ASEAN neighbors, would do well to embrace this biological relativity as well. Since the first One Health seminar, a lot has happened. Our partner, Corporate Biological Systems, built, together with M2M Engineering, a very serious factory in Pekanbaru, producing an array of micro and macroorganisms. Mr. Jonathan Koppert, here, can tell you about the many solutions his company already offers here in Indonesia. The no-self technology has been tested by state-of-the-art Indonesian research organizations. You will see some examples here today. Also, the interest of both commercial and state-owned companies throughout the ASEAN region is high and robust. Today, you will get a lot of information, perhaps too much to digest in one go. My colleagues are here to answer questions, and what's more, the Italian company NoSelf SRL, established by Professor Mazzoleni, Dr. Diano, and several others, will incorporate this year in the ASEAN group of companies. In this way, we can cater best to the needs of both farmers and patients in this geographic area. We are open for business and for scientific cooperation. We expect many diverse, affordable, and accessible products entering these markets soon, for which we certainly need the help of visionary politicians, regulators, and authorities here present. I very much like to thank Pak Taco and his team for the impressive organizations of the seminar. I hope that more and more younger people will embrace our novel ideas and carve out a future for themselves in the best interest of the many. And a personal message to Professor Bungaran: I dearly miss our chats. Thank you.
Okay. Thank you, Peter. Hope you get well soon and can join with us for the next event for One Health promotion. Ladies and gentlemen, I should apologize because just got the message the representative from Ministry of Health still in Batam. So we will miss this representative. Then I should move to Professor Dr. Niluh Putu Indi Termayanti, who already joined with me here as the head of Research Organization for Health, Agency for Research and Innovation, National Research and Innovation Indonesia, or BRIN. So I won't read the CV because we have run out of time. So just please, Bu, you got about 20 minutes to share what the policy in research and innovation that might be related to the One Health concept, because you are coming from the research organization who deal with health issues. The time is yours, please. Okay.
Can I stand and with my mic? Sorry. Okay, good morning, ladies and gentlemen. Can you share my slide? Okay. Thank you, Mr. Moderator. I would like to present with topic research and innovation focusing on to mitigate and future emerging diseases. I'm chairman of Research Organization for Health in BRIN. Sorry. Ya, ini di sini ya. Ini kan ya. Oke. As we know, the pandemic is still ongoing, so we really understand that has underscored the continued threat of newly emerging and re-emerging pathogens, and a critical value of research and pandemic preparedness effort and a national resilience of health. And we know, as we know, the emergence and re-emergence of infectious diseases continues to threaten of the health of the people worldwide. Sorry. Okay. So what we can do for the pandemic preparedness plan for the next pandemics, for the future pandemics. So there are three goals we want to plan. For the first, for how to we can increase the research. So especially for the characterize the pathogen of a concern and the surveillance to identify threats before they emerge. And the second one is how we can sort the timelines between pathogens emergence to outbreak onset and authorization or approval the candidate diagnostic and medical countermeasures such as therapeutics and vaccines. And this is the third one is very important, how to eliminate existing gap in research infrastructure and technology and expand preclinical and clinical testing capacity. The pandemic preparedness plan goal built on a foundation of the pathogen-specific research that include advancing research on priority pathogens known to or having the potential for emerging as the public health threats. We have, for the pandemic preparedness plan, we have the epidemiology, infrastructure, technology, and coordination. So, if you can see on the slide, we have many experience with the disease. The outbreak have the 5 stages before they can transmit it between human to human transmission. So, for example, for rabies, this on the in the stage 2, so only the disease only infected in human if the human can infected by bit by a dog or cat that infected by rabies. And the other hand, for example, for HIV, the disease on the stage 4, so no needed animal against the disease can transmitted between human and human transmission. Sorry. Okay. As we know, 70%, more than least 70%, the emerging or re-emerging diseases is zoonotic origin. So the zoonotic donating the disease has emerged from an animal and crossed the species barrier to infect the human. Emerging and re-emerging diseases, we know are responsible for a significant proportion of the infectious diseases outbreaks that have the plague humanity over the age. For the impact, what the impact of the emerging and re-emerging diseases? Mostly we have the 3 impact, not only for the global mortality and morbidity. We know have the impact. The second one is the economic burden, and the third one is the social and geopolitical implication. On the slide you can see, I think it's not very clear in here, how the economic burden economic impact of the selected infectious disease during the outbreak. Okay. How to preparedness for the next outbreak or next pandemic caused due to zoonotic viruses or for the emerging or re-emerging diseases. For the best way for future preparedness for the zoonotic viruses and to stop the appearance then reappearance of zoonotic, need to understand how we can understand for first time the primary host, the primary host, and we must know research related the host strains, and then what's the virus spillover event, and the frequency of the infecting the one host to another, then what's the kind of the immediate host. This is the framework there are 6 steps how we can do for the future preparedness for a pandemic. Step 1 is engage all stakeholders, and then human and animal surveillance systems, and then increase regional clinical capacity, and the next step in the step 5 is establish multidisciplinary data management and analysis times and analysis teams, and then engage government and international stakeholder. So the framework for how we can leverage the capacity and capability to zoonotic disease surveillance, especially in remote rural areas, that will facilitate integration into existing surveillance system. Especially for the pandemic or outbreak caused by virus, I think vaccine development for emerging infectious diseases is very important. So, in how we have many experience. How do we implement using the One Health approach? For example, in the H5N1 outbreak in Indonesia, we have the nice collaboration with many stakeholders to control of the disease. We work together with the university, with the Ministry of Health, with the Agriculture Ministry, and another ministry. Okay. So, how to mitigate the risk associated with this yet unknown pathogen? The study will prioritize on the preparedness research on a prototype pathogen, and which are select pathogen identified from viral families known to infect human. And through the target basic and applied research on the prototype pathogens from each viral family of the concern, the accrual of a solid foundation of the knowledge and medical countermeasures, MCM, such as therapeutic, vaccine, monoclonal antibodies, will enable with the rapid response when a novel pathogen from any of the viral families of concerns. What the continue the preparedness of the response, the preparedness research effort will provide the region roadmap of the product development and evaluation of scientific infrastructure and study capacity needed to robust research respond to future pandemic. So So obtaining in-depth knowledge and development MCM for the prototype pathogen within viral families that pose the highest risk for the epidemic or pandemic. So the preparedness for major infectious disease, as we know, will save lives, and rapid deployment of the effective diagnostic vaccine and treatment can contain an outbreak before it's expand to a larger pandemic or outbreaks. And especially for the priority, how to priority and pathogen research. So we refer to this priority pathogen, and this pathogen specific research priorities viruses are most likely to cause a significant human morbidity and mortality. This one, how the research will conduct. We first time do pathogen selection, and then to continue to basic the preclinical translational research, and the next to clinical research, especially for the drug and vaccine or therapeutic. The pandemic preparedness plan will support critical studies to how to characterize a prototype and a priority pathogen, including understand the viral biology and structure, host immune response, mechanism of immune evasion, disease pathogens, and studies to develop assay and animal studies. And fundamental knowledge of pathogen biology, structural properties, mechanism of transmission, including how we can identify the viral vector, viral life cycle, viral entry mechanism, and host receptor, tissue tropism, and host immune response is critical to effort informing the development of medical countermeasure and against new re-emerging or emerging pathogen. So water BRIN can support to conduct, especially to health authorities, national health relations. So BRIN have the research organization for health. We have the 7 research center. So research center for biomedical, center of biomedical research, research center for preclinical and clinical medicine, research center for public health and nutrition, research center for pharmaceutical ingredients and traditional medicine, research center for vaccine and drug and Ekman, research center for microbiology, and research center for veterinary science. And the other hand, BRIN provide for the all research through Indonesia, provide the research funding, infrastructure. The infrastructure belong to BRIN can share with all the scientists through to Indonesia. And we have the human resources. This is research scope in our research center for biomedical research. So if we hope we can make the collaborate with the other scientists in the nation or in the Indonesia or from the board. This one, the research scope under the research center for biomedical about the diagnostic research, stem cell research, and phenotypic, genotypic research related to biofilm, IMR, micro and microbiology, and how do we conduct the population research, especially for genome-wide association studies and laboratory-based disease. And we have the research center for preclinical and clinical medicine. The research center conduct the research related to preclinical and clinical medicine, especially for communicable disease, and then preclinical and clinical medicine of the non-communicable disease and mental health. And the next, we have the Research Center for Public Health and Nutrition. So the research scope is very broad, So including the one maybe as we have the vector infectious disease and zoonosis in a human. We have the research group that the research group conduct the research with the topic of factor infectious disease and zoonosis in human. And we have the Research Center for Pharmaceutical Ingredient and Traditional Medicine. The research scope regarding the research of on the development medicinal raw materials, research on the standardization of traditional medicine with using the raw materials, especially from the origin from the Indonesia, and research from the development of traditional medicine, research on the formulation, and something like that. For the Research Center for Vaccine and Drug, we have three focus on our research. First of all, for vaccine development research, especially we develop the protein platform, RNA and DNA platform. Halal vaccine is it's our focus in our research, and the biological product development research, especially we development of biopharmaceutical production such as the monoclonal antibodies, nanobodies, recombinant protein, and something like that, and drug research, how we can discovery of drug for the communicable and non-communicable diseases from the nature, from the marine, and something like that. Ekman Research Center for Molecular Biology, they conducting the genome diversity, molecular bacteriology, malaria vector resistance and malaria pathogen, dengue, hepatitis, RBC, cytogenesis, molecular therapy and structural biology, and stem cell and organoid cell. This one, the Research Center for Veterinary Science, the scope, the research activities are related the early detection, early report, early response for the animal and zoonotic diseases, emerging and re-emerging disease and zoonosis, bioterrorism, especially to biological risk, implementation of One Health, utilization of veterinary innovation, animal welfare, and veterinary standard testing. Okay, we have the 5 focus research in our research organization. We have the one we call the program home program in our research organization. First, the topic 1 related the drug and vaccine control of the infectious and regenerative disease, and the second topics, development of the research and innovation in precision and regenerative medicine, and and another topic with the development, how we can develop the medical devices and health protection tool, and the other topic we have the infectious disease research and innovation, and research and innovation for stunting reduction through a family approach. This is in our vaccine roadmap research in our research organization. We developed the TB vaccine, HPV vaccine, and then we developed the biological product such as the NH1 gene from the dengue and something like that. Okay. This scheme, how to BRIN can support in vaccine development in Indonesia. So BRIN provide the funding, especially for the in Indonesia, the lab scale, how to vector expression, how we make the master seed vaccine, and till how we production with the CGMP, and then preclinical testing. The funding, the BRIN provide the funding till the preclinical and clinical testing for how that one you can see in the slide, how the BRIN can support in vaccine development. This is for the infrastructure. So now BRIN nearly complete for the animal BSL-3, especially for macaca. So BRIN support by Pak Pak Imung to develop how we develop and build animal BSL tree for the macaca for the preclinical testing for the vaccine and drug, and how we can prepare to mass scale for the vaccine. And BRIN have the genomic laboratory for all the working related the DNA, RNA, and something like that. And BRIN have the CGMP lab consists 2 lines, especially for CGMP yeast and CGMP bacteria. So for the structure and function studies, BRIN have the cryo-EM in BRIN is coming in December 2022 at the genomic lab. So the infrastructure, the equipment can the equipment can share with another researcher through Indonesia to support the technology including X-ray, crystallography, nuclear magnetic resonance, imaging, cryogenic microscopy, and something like that. And I will skip this slide. And in preclinical research, we conduct how to provide the expansion of the specialist technology related the high throughput cell sorting, cytometry, immunologic assay, sequencing, and support the genetic examination of the zoonotic reservoir that might contribute to emerging infectious disease of the global health importance. I will skip this one. Institution, including our institution, BRIN, should leverage its expertise, especially in infectious disease, genomic, proteomics, bioinformatics, and access to clinical samples to develop a rapid response diagnostic for biological threats and emerging infectious disease. And especially for the therapeutic, we support basic translational and clinical research effort to identify promising target for intervention and generate novel therapeutics that are both pathogen specific and have a broad activity. So, for example, we development of small molecules such as the monoclonal antibody, and have the provided scientists with the valuable tools to prevent and treat infectious diseases. That one especially for the infrastructure, BRIN have the many infrastructure we share with another researcher, especially for the preclinical and clinical research infrastructure for the animal BSL-3, animal BSL-4 maybe, but we have we have no yet the BSL-4 capacity. For the clinical and preclinical research, preparators includes leveraging and expanding existing domestic and international outpatient and inpatient clinical trial sites, enhancing on-site expertise and developing the clinical research infrastructure needed to evaluate countermeasures and rapidly respond to infectious disease. Especially this is for the if we continue to clinical study.
Please your time, Bu.
Okay.
2 more minutes.
Okay. I think it's my last slide. So we need the effective coordination and communication are central to effective preparedness research effort, and requires a close collaboration and intramural and extramural programs as harmonized and supported by effective mobilization on logistical, administrative, and technical resource. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Ibu Profesor Niluh Putu Indi Darmayanti. Professor Indi, yeah, because in Bali used to be abbreviated by NLP. Yeah, NLP. Niluh Putu, yeah. Okay. Thank you very much for a very thorough presentation and related to the research and innovation policy in Indonesia. And this is something really new because the agency, the BRIN or BRIN, is close to 1 year, right? Yeah, almost 1 year. Yeah. Well, first,
And for the for the research organization program,
yeah.
6 months.
Okay. Okay, thank you. Ladies and gentlemen, we have heard together how the complexity of health problem in the world, and it's come to the conclusion that One Health is one of the solution to how handling this problem. And Professor Indi also, some of the, on her presentation also mentioned about how the importance of the development of therapeutic and vaccine in conjunction with health remedy. And I think we will hear this more from our next speaker, but we are now having about 15 minutes for question and answer, or I think that if we will put together with the second speaker, because we will manage the time as the representative from Ministry of Health is not clear yet. So I would like to invite Professor Stefano Mussolini to come with us here. He's a professor from University of Naples, Federico II in Italy. Go ahead, Stefano. You got also 20 minutes.
It's already on. Okay. So, thanks very much for. I'm really honored to be here for the second time, and I wish to thank the organizer for inviting me, Professor Bugaran especially for addressing the welcome speech for the second time, and I'm really like to be here again. I prepared a presentation. Can I have my presentation? Sorry? You find it? Yes. No, it's not this one. No self-concept. Yes. Okay. So this is the title of my presentation, introducing you to the possibilities of applying our idea, conceptual no self-concept, to application in agriculture. You have seen in the brochure that has been distributed to all participants this summary picture. And essentially, this is the concept of using the DNA of a pathogen species, of a parasite, fragmented as the inhibitory product to control that pathogen. This concept is easy to explain, as it is in the figure, but it looks strange, I would say. Okay, it's not intuitive why the self-DNA of a pathogen should limit the control of that same pathogen. So I like to start with a very, very general view that shows you my former background is ecology, plant ecology, and I like to show you how I arrived to this discovery and to understand the possibility of applying in the way that I showed you before, okay? So let's start with a very, very simple concept summarized by this statement: DNA is everywhere. What do I mean with this? If we move to a tropical forest, you have big biodiversity, lots of different trees, species, animals, microbes, insects, butterflies, everything is here, okay? And if we move, if we zoom in the forest, and you can actually think to go on a single leaf and to see a little lizard that is on that one, and the same leaves can have a cricket or a fungus growing on that leaf, you can imagine that each of these organisms has its own DNA, the self-DNA of each species. That's an obvious concept, okay? And you can imagine in a big forest how many different self-DNA for every species are there. But then the organic cycle, the life cycle, all the organisms also die, and they will fall on the forest floor, and they will start decomposition, and their DNA is still there, although if they are dead. And they start decomposing, and their content, the DNA that was in their living cells, is going to be released into the environment, into the organic matter that is in the soil floor, in this case. If you zoom in and you look at the microbiome that is in the soil, all the microbes that are there, these microbes are actually living in the water, in the organic matter, and are surrounded by all the DNA that is coming from all the organisms that are in that system. But the same microorganisms have a very high turnover. They reproduce and die. And every time a cell is dying, it's releasing its content into the system. And what is interesting to specify in this very moment is that when the decomposition is going on, actually the DNA molecules are the most resistant to the decomposition processes. So they stay in the soil in this case, also when all the other organic material compounds have disappeared and have been digested and recycled. And I say to students, smiling this, saying, think of those movies where the investigators find the killer DNA after many years. You know, it's the same concept. The DNA is very resistant. It can stay for a long time in the environment, okay? Now, what is interesting to understand is that all this environmental DNA is part of the nutrition of all living organisms. So plants, while absorbing nutrients and water from the soil and things, they also absorb the DNA that is present in the soil, the free circulating DNA. And this happens also to animals. When we eat food, okay, you have a pasta dish, I'm Italian, so I like pasta, you will have DNA of the tomato and of the wheat and of the olive that you use for cooking it, okay? So DNA is everywhere. And if you think of the trophic chain, all the animals and plants are then connected, and they exchange this DNA through the trophic network. So in the view that I'm presenting, when you look at a complex system like a forest, you have also to remind that in that system, the life cycles is circulating DNA everywhere. If we go back to the forest view then, it should be clear now that there is a big system dynamic going on, flows of everything. I'm just saying, remember that it's not just flows of water and nutrients, but also of DNA. We should not be scared by this concept. DNA is a very natural molecule part of this life cycle. But what happens when we transform the landscape to agriculture? We start removing the natural vegetation, and the land becomes very different. And when a big intensive agriculture system is applied, then you have one species only in substitution of the original highly biodiverse ecosystem. Then think about the accumulation of DNA in this case. And in this case, the DNA will be coming from only one dominant species, okay? So the view in terms of DNA circulation is going to be different. In a natural system, there is that network, very complex network of trophic exchange of DNA through all the organisms. In this case, there will be an accumulation of the self-DNA of the crop. And we are aware that if you, especially in dry conditions, you repeat the cultivation of monospecific crops for many years, then the productivity is going to decline. In Europe, we are very well aware that there is the so-called soil sickness that is not just a nutrient depletion. If you fertilize with chemical NPK fields that have been cultivated for too many years by the same crop, you will not solve the problem. And the ancient Romans, they more than 2,000 years ago, already wrote in their text about agriculture that rotation concept between crops was a good concept. In fact, when you have rotation between different crops in agriculture, in traditional agriculture, then of course there is not accumulation of the self-DNA of one crop, because you will move the crop in different places in the field, okay? So expert farmers know that it's a good practice in traditional agriculture. What is very interesting to mention, being in Indonesia actually, is that also rice paddy is an interesting case to understand, because with this type of cultivation, with the flooded by water, you can keep the monospecific growth forever. So you can have rice paddy that can be hundreds of years repeated every time without problem. Then why? Because DNA is soluble in water, is washed out, and so in this type of system, you don't have the accumulation of the self-DNA of one species only. You have the water flow that clean up the system continuously. So what I'm saying, this is a new view of this problem, is that in dry land, you have sustainability only with biodiversity. If you get monospecific crop in dry land, you face a problem. When you move to water system like rice paddy, you can not worry about, okay? That is sustainable also if it is monospecific. Now, let's come to no self-concepts. With this background, what I showed you before was my background, studies in agriculture and organic matter cycles and thinking of this DNA concept. I came up to this concept. Okay, let's have litter then, organic matter. You decompose it, and at the end of the decomposition, you have the humus, and everybody knows, also people that are not expert in agriculture, that humus is good for plants. So the plants can grow on the decomposed organic matter material, okay? We studied a lot this. I'm not going to give you the details on this. We did the chemical composition of fresh leaves and decomposed leaves, okay? And it changes, of course, because there is a lot of processes, biochemical changes going on during the decomposition. But what is the very important point is that the fresh materials, sorry, what happened? Okay. I'm saying that the fresh materials are not so good as nutrients for plants, the humus, the decomposed material, are good for plants. But if you think of microorganism, the microbiome, it's the opposite. The fresh material is good food for the microbes that like to decompose it, and the decomposed material, the humus, is stabilized, stay in the soil, is nutrient for the plants, but not good anymore for the microbiome. So you have this concept that fresh material can be phytotoxic, stabilized material is a good fertilizer. This seems a very, very general concept. But I got this idea more than 10 years ago. I told you this also last time that I was here. That through the decomposition, it's true that you create humus that is a fertilizer, the blue arrows, but you also release the DNA. And if that DNA is the self-DNA of that species, then that can be inhibitory. And now we arrive to the point. Forget about that. We studied funny plants like these little plants dying in the middle or the regeneration in the forest, but these are details. If you want to discuss more, I can explain you more. And now let's stay with the story at a simpler level. The discovery was this one. If we put plants in presence of heterologous DNA, of DNA of different species, they grow very well. If you put the plants in the presence of their self-DNA, they are inhibited, no germination and no growth. Okay? That's a very simple experiment, can be done by everybody, on any species. It works always on any species. And if you check on bacteria, the same. And you do it on protozoa, the same. The self-DNA is inhibitory. And if you do it on algae, we observed with Marcello Diano the same effect. And fungi, the spore germination is inhibited by the self-DNA, but not by the DNA of the other species, okay? And in insects, sorry, it's quite impressive. If a larvae of an insect is eating DNA from a food for it, there is a complete metamorphosis and completion of the cycle. But if the same larvae is eating itself DNA, the cycle is stopped, is inhibited. And now this concept is quite amazing. We start working then on model organisms like Arabidopsis in plants, and we start publishing things. It's very interesting. The root of the plant, in presence of the self-DNA, does not absorb it. The fluorescence that you see here is the DNA. It remains outside, and the plant becomes impermeable. It's not efficient in nutrient uptaking, and inside the cell, you don't see evidence of DNA uptake. Whereas, if you put in the plant non-self DNA, you see the fluorescence going inside because the plant is uptaking the non-self DNA that is a good food, and inside the cell, you see a lot of metabolic activity that is stimulated by that. So it's a very, very clear evidence on plant, on Arabidopsis plant. But then we start working on nematodes. We collaborate with the CNR Institute, the National Research Institute in Italy, and we get very clear results of very good life of the worms in the presence of non-self DNA and inhibition of egg and larval arrest in very significant results in presence of self-DNA. Ongoing results on Drosophila melanogaster, just to show you that the experiments now are very robust and tested on many, many different species. And very clear results. You can see by eyes, even if I don't make any comment on that. So, let's make the story very simple. All organisms in presence of an environment rich of non-self DNA, of DNA of other species, they are happy, okay? They are healthy, let's put it this way. But if you enrich the environment with their own DNA, then they become unhealthy, they are weakened. So this is the concept, what I call the no-self concept. And now we go to more in depth on the subject of this conference. If you think of the traditional phytopharmaceutical research, you start from any sources, you extract molecules, yes? Okay? And, or you can have molecules to be tested by chemical synthesis, that's okay. And you have thousands of molecules that can be searched and tested by random screening to search for their bioactivity and possibility of using them as drugs. But through this process, you arrive to have actually an approved drug in a long time. This process can take more than 15 years of research, and there are problems. Some of these molecules can be, the process is very long and expensive. There is not specificity of actions, and these molecules can be toxic not just for the pathogen, but also for others. Like if you use antibiotic, okay, the antibiotic is not good for the health of all organisms, not just for the microbes that we want to kill. And most problems comes from the appearance of resistance problem. Because in this case, if the resistance appears, this drug cannot be used anymore, and all the investment is lost. So we come up with this idea, very simple. Forget about this. It's a bit strong to say like that. And eliminate all the problems, and just take the DNA of the pathogen and use that DNA as the inhibiting product. And we say that this can be the solution. 1 year only to do all the process, and a huge advantage in terms of R&D development, time and cost. Now, if the concept is clear, imagine a new future for agriculture. The traditional approach, if you have a pathogen here, this worm, the black worms, killing the crop, we spread synthetic pesticide. It's true that we kill the worms, but we also have residuals in the crop by the chemicals, and we also create damage to the ecosystem around the agriculture, okay? In our concept, we change the concept. We take the DNA of the pathogen and use it as the inhibitory product, and we get a healthy ecosystem with the plants that are very healthy in this condition. And in medicine, imagine that you have antibiotic treatment of microbes that is effective. Okay, the guy is happy because he's recovering. But if the pathogen is antibiotic resistant, then there is a problem because the antibiotic don't work. And we say, okay, let's test if the self-DNA of those pathogens can be used and cure the patient. So it's the same concept. And now we come to the interesting part of the story. Okay, already what I said was interesting, but that all organisms have a microbiome, okay? It's funny to see a plant compared to a man, but a plant is a living organism that uptakes nutrients from the soil using the digestion, the decomposition of organic matter that the microbiome does in the soil. It's similar to what happens in an animal. The only difference is that in animals and men, the microbiome is inside the gut. But still, it decomposes, digests the organic matter that is the food. Okay, it happens inside, but it's the same concept. So digestion is similar to decomposition in a soil. And why I'm saying this concept? I show you a very, very nice example that was told to me by an organic farmer that runs a very, very nice farm. He's crazy about organic agriculture. He would never add a chemical in his farm, okay? And he told us this story. His cows are fed with trifolium hay, very high quality, that he produces. And he takes the manure of his cow to fertilize the tomato and gets very good tomato. Then he uses the same manure to fertilize the wheat, and he gets very good wheat and makes wonderful bread. But then he used the same manure to fertilize the trifolium hay, and he got a problem, the soil sickness. Now, with my explanation, this is clear why. The DNA of this is going through the gut of the cow, is handled by the microbiome that is in the gut of the cow, and in the manure, this is enriched by the DNA of the trifolium. And so, this DNA is a part of the nutrient cycle of these other crops, but it becomes negative, self-toxic to the trifolium. So, this is an anecdotal story. It's not science. It was told to me by a farmer, but it represents the way we went on, okay? We started working on this concept, and now I'm going to closer to the conclusion. Imagine then, in human medicine, you get a gut, and in the gut you can have a healthy gut, high biodiversity. Lots of bacillus, lactobacillus, good organism in our gut, making digestion of all the food that we eat. And we know that if our food is diverse, it's good for health. But if you get in a dysbiotic state, and you get a high level, for example, of E. coli in your belly, then is unhealthy conditions, and this can be treated with this concept, DNA extraction of this red pathogen, and using it, provided by a natural library of yeast, for example, and then you bring back the dysbiosis to the balanced diversity of the microbiome. So, it's the same concept again in a different situation. And now we come to the bridge for the next communication that Marcello will give. So we thought, okay, the concept is very powerful. We got an idea, using the DNA to control a pathogen, the self-DNA of that pathogen to control a pathogen. But how do we produce that? How do we distribute that DNA in the soil, in the environment, in the food, okay, according to the conditions? And so we start thinking, okay, let's use algae, and Marcello will tell you much more about that later, and the algae are known to be biostimulant on the plant. If you give algae in the water, in the irrigation, the plants will grow very well, okay? But then, let's give to the algae the DNA of the same plant. The algae will grow very well, identical to the other case, because they will add that DNA that is non-self for them to their uptake, but then the same algae becomes inhibitory on the same plant. The only difference between these two productions has been that this stage, they were exposed to natural decomposition of this plant material. So this microbiome, this algae, became carriers of the DNA of the target species. And now we come to the upscaling concept, and Peter Jens mentioned that with Koppert we already started a production in Pekabaru doing algae and upscaling them and things like that. So you can imagine a pathogen given to an algae in some, sorry, not the pathogen given, the DNA of the pathogen given to the algae, and then you grow the algae, upscale them in the big facilities, and then you get this fertilizer product. But that fertilizer product is special because it will be dual use. It will be fertilizer for the plants and controller for the pathogen. So a dual use microalgae. And now we go to the end of the story, and I will show you a last example on oil palm case here in Indonesia. This is the traditional view. Organisms need nutrients. That's true for plants, for animals, for humans, okay? But then there are the pathogens, and we want to kill the pathogens. That's the traditional view. Every time there is a pathogen, we want to kill it, okay? That's understandable. We add this concept. We must be aware that the health of the organism depends on the possible accumulation of its self-DNA in its environment. And the weakness of the organism can enhance the pathogen. This is the no-self model idea, okay? We can change this by adding microalgae to the cycle that are inhibiting the pathogen and biostimulating the organism at the same time. Now I show you, and we think that this is really a One Health approach. Okay, but I will tell you a little bit more on this before concluding. We did, starting since the last time I was here in Bogor, some work on the oil palm case, and we simply demonstrated in the lab a very simple concept that now you should follow me because I told you already several times. If you add to oil palm the self-DNA of the palm, you can get inhibition of the growth of the palm, okay? Because the palm doesn't like to grow with its own self-DNA. But also, if you take Ganoderma and you grow Ganoderma in a plate, in a petri dishes, if you add the self-DNA of Ganoderma, you can observe inhibition of Ganoderma. So I'm repeating the same concept. The self-DNA is inhibitory on the same species. And now what's happened? I took this picture 3 days ago in Peccabaru. Oil palm plantation, thousands of hectares, monospecific DNA accumulation. The palms in first cycle of plantation have no problem. Second cycle, so-and-so. Third cycle and fourth cycle, they are very much affected by Ganoderma. So the old plantation that are more than 30 years repetition of crops, they start to have the Ganoderma problem, okay? And with all the things that I've been telling you, what do you think now of the practice of adding the empty fruit branches around a single tree to provide organic fertilization? That's a good attempt by the farmer. But in my view, you are creating a high concentration of self-DNA by the decomposition process for the palm. That's not good for the health of the palm. In the long run, of course, not immediately. Immediately you can get an organic fertilization. But by the accumulation of the self-DNA of the palm, you create a negative condition. And in that negative condition, you get the Ganoderma attack that can kill the plant. So in summary, it's just the last few slides, what we are saying that here is a palm, the decomposition of the residuals, the litter of the empty fruit branch of the palm is releasing the self-DNA of the palm, and if this becomes too much, this is creating inhibition on the palm root system and facilitate the entrance of the pathogen attack. And the Ganoderma then becomes happy and growing in that conditions. And remember the microbiome concept. What happened to all the microbiome that is in the soil? If the soil becomes full of self-DNA of the palm, the microbiome will be also an additional transmitter of the self-DNA of the palm. So it is a vicious circle that creates negative condition for the palms. And now, if we take the DNA of Ganoderma and create a natural library of algae containing the DNA of the Ganoderma, then this product can be grown and becomes a dual-use product that is a biostimulant for the palm and an inhibitory for Ganoderma. Simple like that. Sorry, I was in the wrong direction. Okay, I repeat the concept here. So I think it's useless to say go on. So the conclusions. I've been explaining you now an example, the oil palm Ganoderma model. But in the way I'm presenting it, you should understand that then this could be described for the banana fusarium model. Banana is a big, extensive crops in the tropics, monospecific crops. After many years of cultivation, they're facing very strong attack by fusarium that is becoming a pest. And they're trying to compensate this with searching for resistant banana varieties, okay? But in my view, the problem is the accumulation of self-DNA of the banana that is enhancing the fusarium attack. So we can say the same story that I told you for Ganoderma and palms for that. But then, potato Phytophthora, tomato Robanche, we work in Italy on this, grapes and Peronospora. So we can say this for any crop and any pathogen. It doesn't make any difference. Any livestock and any pathogen of a livestock, a fish, a parasite in the gut of the fish. We are doing research with the vet veterinary school in Italy by adding as food integrator to the fish diet algae with the self-DNA of the pathogen of the fish. And so we get stimulation of the fish and inhibition of the pathogen. That's the concept. It's always the same concept. And human, why not? Of course, humans take more difficult legal issues, but we are working on some institutes in Lisbon and in Napoli, also on oncology and other ideas, okay? And so these are just examples of things, and our motto then is, as nature does, because we think that our approach is not adding a chemical by synthesis, by industry production and things to the system, but is really to modulate the system in a natural way, as it actually does naturally, okay? And the last point, and then of course, if you have any question, I'd be happy to answer, is then when you spray our product, let's say that the algae with the self-DNA of a pathogen, then the day after, of course, you don't see anything, okay? It's not like spraying an herbicide and then the day after you see all the weeds killed, because that's a killer. In our system, nothing happened apparently, but starts a different dynamic, and then in 15, 20 days, the plant starts to feel a little bit healthier, and then the leaves becomes a little bit greener, and the pathogens becomes a little bit less aggressive, and then in some months, the system is rebalanced and becomes healthier. So that's why we feel that our no-self concept is very much a One Health concept, because it's not entering in a... yes, I finished. I was last. My smile. Thanks, David.
Okay, make a big applause to Stefano Mussolini. Actually, I let you over time because it's very important, the subject. So, but still, we have another speaker next to you. But if you don't mind, later you can join here when we have a discussion, okay? Ladies and gentlemen, we have been shown together by Professor Stefano Mussolini how the impact of the One Health approach to mitigate what the complexity in the health problem that we face today or the future. And one very important thing that I note from this presentation is that a monospecific crop in the long term will not be sustainable. And this is very, very serious issues that we need to take care of. Because, for example, we all knew Indonesia is the first oil palm producing countries, and this is handled with the monoculture, okay? But it is something that you mentioned, the old traditional technology like rotation and also organic input may also alleviate the impact of this self DNA effect. This is very challenging and important, but still a question there where we could apply this agent to the target, and will be the subject of Marcelo Diano. Please come with us here about how to develop no-cell technology product using a microalgae as production and carrier platform. Time for you, just okay. You got also 20 minutes, Marcelo.
Hello, everyone. Nice to meet you. I'm Marcello Diano from Italy, from Naples. I'm the CEO of M2M Engineering in Italy. Many thanks for participating and for coming. Thanks for coming to all colleagues, and many thanks also to Bungaran and Mr. Taco, Minister, and Head of Researchers Agency. Very happy to be here again for second time, the first time in 2019. So, we continue to discuss evolution history. How use macroalgae as a technique for production and carry as platform for no-cell technology. Thanks for Professor Mazzoleni to very, very interesting, presentation before. So I continue the history. Just a brief introduction of myself and our company, M2M. I'm CEO. M2M Engineering is involved in scientific research and industrial application of macroalgae since 2000. So it's more than 22 years working in macroalgae with several customer collaboration in several countries, in Italy, in Europe, Sweden, Spain, Germany, Netherlands, Europe, and also in other countries in Indonesia. I'm very happy to collaborate, and we are making a serious collaboration with Koppert and Biocycle. Also, I want to thanks to Mr. Taco to organizing this meeting, to Budi Tanaka, becoming from Biocycle. We are making a serious industrial production with Biocycle and Koppert. Thanks also to Jonathan Koppert to this strict and serious collaboration, industrial production in Indonesia for these developments. We have also several patents in macroalgae cultivation, above all to increase production, to increase production and optimize the productivity and have a increased production in industrial farms. Our experience, of course, we have several field of application, like in pharma, in livestock, in agriculture, also the other research and developments in the other new sector. So just a brief overview of our activities. We follow the interchain from the laboratory strain maintenance. We have also our own strains. So the system for maintenance, strain, laboratory, for research development, producing very strict controlled production. Also, industrial production, industrial photoreactor for production in open field, and of course, all system and all the industrial production needs a strict monitoring and control system to control the whole production. And we are also apply to improve the production, improve the productivity, the biological modeling of macroalgae and interfacing with also the sensoring and monitoring system, also to increase production, maintain full control of the production to achieve the maximum productivity in all places. Several, as I said before, we have several collaboration around several, many countries, also in Indonesia. We are very happy this collaboration is Peter Jens. Very thanks to Peter to organizing and to push on this type of concept. As I agree with Peter, I very like the Indonesian environment, Indonesian people, because it's a very kindly, open mind, open to new innovation. So I think several of you already we meet and discussed in last years. So just very try to be short. General overview the macroalgae. In general, we can view as four groups of macroalgae: green macroalgae, red macroalgae, diatom, and cyanobacteria. In general, macroalgae is a unicellular organism in order of the microns dimension that using sunlight and nutrients and the CO2 from culture, using for growing, so also for production. And difference from seaweeds from the sea, it's algae from algae with the leaf. So multicellular is like similar to the plankton growing on the sea, but differently. Macroalgae is a unicellular organism that growing from the laboratory to so the order, the magnitude of each cell is the order of microns, and the type of chain of the production is different because seaweeds growing on the sea differently. Macroalgae is industrial production with controlled water, controlled nutrient, controlled system, closed system. So the type of approach in production and also harvesting in seaweeds, it's becoming from the sea. So also in harvesting is different type of the harvesting with some system or some also some manually harvesting. Macroalgae instead is different. The type of cultivation is with the industrial production and also type of harvesting is industrial in controlled condition. So we continuously have under control the full production also in type of the harvesting. Type of system used for growing macroalgae can be a photobioreactor is a closed system, can be a different type of system. These are some of our plants realized like a multiflat or tubular, cylindrical, the other type of tubulars, and also multi-flat and flat system, or the other system can be open, like a thin layer or raceway. We prefer use for increased productivity, and for some reason, to use the production in open system, also in open system, but in controlled environment. So in general, we use this type of system, closed, or this type of system, also if are the open system like a raceway or a thin layer, but in controlled and closed greenhouse, so the controlled environment. For some reason, first about contamination, about controlled product, because from rain, from environment, it's a vehicle also of pollutants and some contamination. So in the end, the end product that we produce is very stable and very clean and controlled, certificate, and with the several strict procedure for also for control. So also in open system we grow in closed system also to maintain light under control, temperature air under control, and temperature of cultivation under control. Of course, one of the other parameters that we maintain with the strict control is water. We use clean water with the controlled system to also to purify and to maintain control. So just to summarize the different type of approach in seaweeds. It's in open sea, so we have also the variability of the end product, and also, of course, type of product is different because the one of the problem well known in seaweeds is also the contamination from heavy metals, for example, becoming from the sea, because on the sea you cannot maintain control of the water. One of the important achievements in our product is, for example, the contents of heavy metals. We have 2 orders of magnitude lower than seaweeds, but it's a normal because the type of the industrial production is different, because we use a clean water and control it, and the maximum of heavy metals is, for example, 0.1, but many times, and also I'm very happy in of last analysis, many times we have undetected contamination, undetected heavy metals. The other important point is stability of product. In industrial controlled production with the full nutrients under control, light under control, temperature of water under control. So in the end, during the full year, we have all batch, all product certified with the stable product, with the same characteristic, with the full parameter under control. This is very, very important also for market access, for human, for application, for human, also for livestock, also for agriculture. Maintain under control fully parameters, fully contamination, maintain in stable range, and have always consistency and certified condition. A certified end product is important for also for many type of field of application, industrial product, like in agriculture, like in pharma, like also in livestock. The current application is in health food for human and also in pharmaceutical is also know this type of application. It's already commercial and it's also ready. What I want to also show the new innovative application that we are studying in last at least 8 years. We developed a product for agriculture and livestock that they want to show the interesting things. We developed for agriculture a biostimulant product for seed care application, foliar and drip irrigation. I tried to very short. These are, for example, some picture of course, in the last 8 years, we followed research and development in laboratory to maximize the characteristics, to maximize the biostimulant effect, and to achieve the stable product and industrial product for agriculture. So 8 years, it's we made a lot of things in laboratory, in greenhouse, controlled greenhouse, uncontrolled greenhouse, and also open field trials. Now we are going also the 4th years of field trials in open field trials. This, for example, some effect on seed care application in soy, in rice, for example. These are interesting things, for example, in application in problematic seed. Sometimes you have, you have the some lots or in some condition, some dormancy or problematic seed. In this condition, this is an example of the very interesting effect of the stimulation coming from the chemicals, from the hormones and the compounds contents in macroalgae. Please, next. Okay. So just as I said, we have also several trials ongoing, and also in last 4 years we made a lot of field trials. This is a classical effect, for example, this in seed application in wheat on several hectares in field application. This, for example, in baby leaf, in spinach, in baby leaf in arugula. These are field trials for, officially field trials for, also for registration product. So we are already in a commercial phase. This is an example of the big field trials randomized with the randomized plot on seed and foliar products. Next. This is the other field trials in several hectares on wheat in southern Italy. We are making a trials in several places in Italy, in southern, middle and northern Italy, also in several other countries. I also like to visit sometimes to survey the trials. Next. This is the other officially trials with the randomized plot, big trials on corn. This on tomato, again, officially trials for to have also a statistically for product, for commercial. Tomato. Next. Also, we are applying industrial seed company for seed treatment in industrial chain. So we are already in this phase, also in application in big industry in seed treatment. Other trials in grapes, in Italy, grapes, it's also very important in grapes, table grapes and wine grapes. Next. Okay, but some, just some numbers. The effect is increased yield. For example, we made also several trials in comparison with the other commercial product. For example, in this case, it's a test with three competitors, three products with the foliar application in wheat, and this is our product, biostimulant. So it's also confirmed many times this type of behavior, this type of results on yield. The other field trials comparison with the other seed care product. These are two competitors. This is our product on seed treatment application. Next. I want to just as I said before, the important thing in this type of product that we have a constant and consistency results. The product is very stable, but becoming from the type of industrial production, we have a all batch controlled with the same characteristic, with the same efficacy in the end. We don't have a variability on product. Some numbers, some increase of yield in tomato, in lettuce, this in drip irrigation, this one in foliar application. So, coming to the livestock product, as you saw before in agriculture, the product, the macroalgae product, our M2M product for algae, is a biostimulant product. In livestock, we have we are making we made and we are also ongoing the other trials with the shrimps in fish and also the other animal to apply as health feed supplement. So integrating in diet from the 1 to 5% of the total diet, if you add the macroalgae is in this percentage, you have in general enhances disease resistance and improve the health in animals. So, next please. So, as I said, we made and we are making we have a lot of ongoing things on this with the University of Naples Department of Medicine and Veterinary, and also with the I thank to Dr. Romi also in Indonesia on shrimps. Many thanks for trials that you are doing. I'm very so in general, the effect on chicken, on fish, on shrimp, it's health improve on livestock. So increase stress and disease resistance, and this is a good effect that you can use also to reduce the antibiotics. The other big matter on health of livestock have the immunostimulant activity, better livestock health, and increase survival rate. This is very important because, for example, in hatcheries, you know, in hatcheries, the survival rate in the first stage is very important in production because if you have a lower survival rate, of course you have a lower number of the shrimps to grow, and in the end the production will decrease. So, becoming to the yeah,
3
two
minutes.
slide.
3 minutes.
Yeah, okay.
Okay, go ahead.
Coming to the no-self technology, imagine this macroalgae. I like this this example. As already showed Professor Mazzoleni, coming to the no-self technology and fragments of self DNA of Professor Mazzoleni, imagine macroalgae is a truck. It's a truck with the biostimulant effect in agriculture, in plant, and also in livestock. Image is a truck. And image if, in natural way, we charge a fragments on this truck, and we use this truck as a production and carrier for bringing agriculture for plant, or for bringing human for as a carrier for example, to against some pathogens, or use this to for livestock to improve the health and decrease the pathogen in livestock. So the concept is, as a natural combination as nature does, image the macroalgae grow in medium and substrate with the fragments becoming from this discovery of the Professor Mazzoleni. Give time to macroalgae uptake to these fragments. In the end, you have the truck charged with this fragment. So after this, image use this type of technology that I showed before, this type already industrial, ready for commercial, already ready for production. Imagine this type of technology to produce a carrier for livestock, carrier for human, or for agriculture. Next, please. So, in last year we worked a lot on these trucks, these fragments charged on trucks. We are already in field trials stage on some for example, in this case, it's a problem on Fusarium attached on lettuce. These results on no-self technology with the macroalgae carrier, natural carrier on Fusarium on lettuce. In the right, you saw the lettuce attached from Fusarium. This is a problem on field. It's a very, very big problem because you lost also more than 50–60% of the total production. So, in this case, there is macroalgae with as a natural carrier with the Fusarium fragments from Professor Mazzoleni technology. These are the results. I think is self-explanatory. Because the effect is dual. Is a stimulant effect, as you saw before, the truck is a stimulant. So you have a stimulant effect and also inhibition effect on Fusarium. So you have a boot effect, stimulate the health and decrease the pathogen with this technology. The other, we have many things ongoing on research and developments, some in first stages, some other like Fusarium, or like some weeds in tomato already in field trial tests. So it's We have a lot of things ongoing using this technology as industrial production and industrial final product for macroalgae with this NoSelf technology. So I think I finished the... Thank you very much.
Give applause to Marcelo Diano. Thank you very much. And I would like to have you stay here. Marcelo, please stay here. And I would like to invite Stefano also here. Unfortunately, Professor Nilo already left. Okay. Ladies and gentlemen, we'll have a very short question and answer. If you have one, as the latest, we have here from Marcelo that the commercial production of microalgae is available right now. So this is also some opportunity. We are in the tropical area with a lot of sunshine, a lot of water, but we haven't have any success production in such product like that. And this is coming the technology that developed by M2M from Italy. Well, let's have some participants to have a question, please. Identify yourself and to whom the question is towarded, and then please make it short because we will entering the lunch break already. Thank you. Okay, the gentleman on my left here.
Thank you, Mr. Moderator. I have a question to Professor Stefano. There's a very interesting presentation, Prof. And I have two questions for you. First question is, do you think that we need a specific amount of the self-DNA to work well? I mean, to inhibit the growth of this pathogen. And the second one, I don't know whether you already used this concept to explore about the self-RNA instead of the self-DNA. Thank you. The second one, I don't know whether you already used this concept, go to the RNA instead of the DNA. Thank you.
The first one?
Go ahead.
Yes.
The first question is whether we need specific amount of the self-DNA to inhibit the growth of the pathogen.
Yeah. Well, in vitro, we observed effects with higher concentration than the one that we observed in the field. That was counterintuitive, because in a petri dish, for example, on plants, we could see an effect of inhibition with 200 parts per million of concentration. But when we use it in the field, we observed the effect also with much lower concentration. And the understanding of this strange result was because of the microbiome amplification effect that I sort of try to explain. We are studying very much now the process of how the microbes actually uptake the DNA and eventually can amplify it. So we can provide as a product lower amount and actually observe higher effect by the interaction with the natural microbiome processes. This is under investigation. About the second one, DNA and RNA differences. RNA is very mobile, whereas DNA is very resistant. So, in principle, you can use RNA, for example, it is used for RNA interference approach, targeting specific genes, but then it must be protected by RNA model somehow, otherwise it degrades, doesn't get the target, doesn't reach the target. With our DNA approach, it's a very robust technique because the DNA is going to be there. It has a shelf life in the stock in the farm for a long time, but also in the soil. So it's an easier tool.
This answers the question. If you're not satisfied, you get your minimum of the last 3 minutes. Okay?
Okay, thank you, Pak. My question is, how did the self-DNA work? Yeah? Is it work for family or genus? For the example, we applied to same graminea, for example, rice, or to another organism. Is it still work for self-DNA? And second question is, I curious about the consideration to using of microalgae. Why you not use the fungi or bacteria with the same with the same ability to improve plant health? Yeah. Thank you.
I understood the second
Maybe what? Or Stefano? Yeah.
If you ask them if we can use other organisms to do the effects, yes, that's the answer is yes. Is not only the algae. We use the algae because we have all the know-how, the technological know-how how to produce them in upscaling industrial level. But of course, we are also working on other carriers, potential carriers that could be fungi or bacteria. And in that case, the upscaling should be done with different technologies. I don't know if this was the question.
Okay. Is there anyone? Dr. Kamarudin, please.
Thank you for to give some inhibition action of DNA. It should be complete of DNA or particular part of DNA, because we will have a problem for production of whole DNA in a large amount in industry. Thank you.
It's a very good question. The basic technology is based on the whole genome DNA that is extracted from the target proteins, but then is fragmented in random fragments. The size that is relatively small, between 100 and 10,000 base pairs. So it's a big mix of fragments of different size and randomly broken by supplication or by other processes. Or by the decomposition in a natural way. When you decompose the organic matter, the result is the same mix of fragments randomly fragmented. So with the traditional approach, we don't know which are the active fragments, if there are some fragments that are more active than others. Okay? But currently we are now working on selection of sub-region of DNA and checking if there are variability of the inhibitory effect using this sub-portion. But this is more, how can I say, molecular biology application to improve the efficacy. But the efficacy is already very high using this very basic approach. Because we get, in the case of nematodes, 100% larval development stop in 2 or 3 generations of the nematodes. So when they are fed with their self-DNA. So it's a really very strong effect. Of course, we can keep working on improving the selection of DNA regions and making it more efficient.
If I cannot, just issue this. It's connected also to a question from Romy. In the real field, it's like a copy of nature, like to this phrase. And the amount need, the effect is very high in field, and the quantity need, it's also very low in field. So already now, the amount, for example, used in field trials that I showed, it's a very low amount for hectare of treatment, for example. We are saying about some hundreds of grams. So also also taking the full genome without any other things. It's very effective and the quantity needed is very low. Is what I.
Okay, okay, the last one, please.
Okay, thank you. So I have questions for Professor Stefano. So basically for the self mechanism to happen, the plants or the organism have to be able to absorb the DNA, is it correct? And after the organism absorb the DNA, I would like to understand what happens after that. Is the DNA integrate to the genome, or is it similar to the RNA vaccine, for example? I mean, is it going to be permanent, or is it going to be just for several months? And after that, I also have a question for Marcelo. I think I can understand about the inhibition happens by the self mechanism, but I am trying to understand what happens, how is the mechanism for the microalgae to be able to boost the growth of the plants? Is it just because we add the no-self DNA, or is it because of the microalgae can produce something that can boost the growth? For example, is it because the algae contain protein or something? Yeah, that's it. Thank you.
This is a very good question. Now, the question on how is the uptake mechanism. First, remember that the uptake is only for the non-self DNA. It's not for the self DNA. The self DNA does not enter the cells. It lives outside and it acts as an emission at the membrane level of the, for example, of the roots or the cells. But when we have an algae, for example, uptaking the DNA, the non-self DNA, the DNA of pathogen, that this is probably incorporated into the genome. We say probably because it remains stable for many, many generations. The natural library that Marcello has prepared and has been tested, they can be grown in bioreactors for months. So going over many, many hundreds of generations of cycling, they are still inhibited. So they contain the self-DNA of the target that remains there. But when I say probably, it's because we are doing work now on long sequencing the DNA, searching for these fragments that have been incorporated. And it's very difficult work for bioinformatics teams. Because what we have understood so far is that the mechanism is not regarding genes, coding regions, but is somehow hidden in the non-coding part of the DNA. That is a massive portion of the genome, but is much more difficult to detect in terms of changes, because probably you know that it has been termed junk DNA for many years, this non-coding part of the DNA. So somehow it's a totally new phenomenon that we are now trying to address, and we require help by molecular biologists and biochemists and organic chemists, and we are working with NMR technologies to find these. We are now finding the first indications, but are still ongoing research. It's not a definite question, okay? But it is stable. It stays there for a long, long time. Okay?
Before I closing the session, there is some some scientists doubt that this cell technology is basically is RNAi. RNAi. Is that true? And then, is there any other groups who's developed this technology in anywhere in the world?
Well, I don't know about the second part of the question. There are groups that I didn't understand what they
Well, maybe in Europe and the United States or the other part who develop similar approach of this cell technology.
No, I wouldn't say so. Of course, there is a lot of work on RNA that is conceptually completely different. Because in that case, you use either cDNA or RNA protected that is actually the same sequence of the gene that you want to silence. So the concept in that case is that you have a target gene, a specific gene, in a cell or in a particle or in an insect, and you deliver that RNA or the copy of that RNA, and you obtain a silencing of that specific gene. But then, since you are acting against a single gene, it's very easy to get resistance in the treated population, because the population can adapt, and you can get individuals that are resistant to that action, and then you have lost your technology. In our case, since we are working with the random fragments of a very broad spectrum of the region, the effect is not against a single gene, but is an overall silence of the whole metabolic activity of the cells. So we've done a lot of work in this direction. And for example, in the cells that are inhibited by cell-DNA, you see a down expression, a down regulation expression of thousands of genes that go down. And the amount of RNA that is produced is very significantly reduced. So you really observe a wave of reduction of metabolic activity by the inhibition. In that sense, I feel quite confident to say that resistance cannot appear, because it's not going to be focused. So it's a very different approach from RNA interference.
Thank you. I think with this answer, we will adjourn our first morning session, and I will give back to Anne what should we do after this. Then let's make a big applause to this our outstanding. Thank you very much.
Thank you, Professor Vijay. I think we've had such very good deliberations in the morning, and this adjournment calls us for some, I don't know whether it's PFA or food or whatever, but it's available outside.